Hello, and thank you for joining us today for BioTek's webinar event. My name is Lindsay, and I will be the moderator for today's webinar. The audio for today's webinar will be coming from your computer speakers, so please be sure they are turned on and the volume is up. To ask a question, please use the chat feature. It is located in the bottom left-hand corner of your screen. You can send in your questions at any time. Our speakers will address the questions during the Q&A session at the conclusion of the event. If we do not get to your question, we will do our best to respond by email. Today's webinar will be presented by BioTek and NovoSib. The speakers include Wendy Goodrich, Application Scientist at BioTek Instruments, as well as our guest speaker, Dr. Larissa Balakrova, President of NovoSib. With that, I'll turn things over to Wendy to get us started.
Thank you, Lindsay. Today we're going to highlight BioTek automation and challenge testing of a novel freshness assay that provides an easy-to-use quantification of inosine monophosphate, inosine, and hypoxanthine as a percent ratio in animal muscle tissue. This webinar is designed to present the highlights of 2 new application notes available on biotek.com. Artfully prepared sushi is a hallmark of Japanese cuisine, and most people equate this food with freshness and unique taste. The Japanese word umami is used to describe the essence of delicious taste often associated with foods like sushi and sashimi. In 1985, umami was officially recognized as the scientific term to describe the taste of the amino acid L-glutamate plus 5 ribonucleotides such as guanosine monophosphate and inosine monophosphate. The highest levels of IMP, inosine monophosphate, are present during a window of time in the early postmortem ATP degradation pathway in fish and meat tissue. Composed of a ribose ring with an attached adenosine group and distinct hydrogen triphosphate bond, adenosine triphosphate, or ATP, is naturally depleted postmortem through a combination of autolytic enzyme and bacterial action that reduce the hydrogen triphosphate and adenosine bonds, sequentially degrading ATP into 5 smaller nucleoside and purine end products: adenosine diphosphate, ADP, adenosine monophosphate, AMP, inosine monophosphate, IMP, inosine, INO, and hypoxanthine, HX. These 6 parameters make up the freshness pathway in animal muscle tissue. Expressed as a K-value, freshness of fish tissue was first quantified by the Japanese team of Sato et al. In 1959 using an equation that included all 6 freshness pathway parameters. This equation was later abbreviated by Karub et al. In 1984 to a 3-parameter equation. Percent IMP and percent hypoxanthine can be expressed as an equivalent freshness index by their relationship in Ki, such that the higher the percent IMP, the fresher the fish. The decomposition rate and concentration profile of the freshness pathway can differ from species to species. Because the catabolites are produced early postmortem, their measurement in animal tissue can be used to determine parameters such as muscle age and storage time, important indicators of quality. The quantity of these nucleotides in tissue products can also influence sensory attributes such as taste and texture. It is on these principles that the NovoSib Preci Freshness Assay was developed. Here to tell us about the assay is Dr. Larissa Balakrova, President of NovoSib.
Yeah, thank you, Wendy. So purine and pyrimidine nucleotides are involved in numerous cellular processes. They provide living organisms with the energy. They are building blocks of nucleic acids, and they are an essential part of cellular metabolism. Animal muscles are particularly rich in ATP since this purine nucleotide provides the energy for muscle contraction. And immediately after slaughtering due to respiratory arrest, as Wendy said, ATP breaks down, leading to the formation of IMP, inosine, and hypoxanthine. These 3 ATP catabolites are important for human and animal nutrition for several reasons. Nucleotides constitute objective indicator of freshness, reflecting the quality of fish and seafood. Nucleotides provide the organism with nutrients essential for immune and digestive systems. And a nucleotide AMP is responsible for good taste of fresh seafood products known as umami taste, as Wendy said. Moreover, for aquaculture and farms, AMP is known to be feeding stimulants. Seafood is high protein food, low in calories and low in saturated fat. So it is a rich source of omega-3 vitamins and minerals and has numerous beneficial effects on human health. However, seafood is also a highly perishable product, which is transported nowadays long distance before arriving to endpoint consumers. That is why the need for rapid analytical techniques to measure the food quality and freshness is greater than ever. Sensory evaluation is still considered as the most effective technique to assess fish freshness. Sensory evaluation consists in observing, smelling, and touching the fish. During this evaluation, whole fish is assessed for appearance, eyes, gills, and skin odor. The advantage of sensory analysis is that it allows rapid evaluation of whole wet fish. However, the assessment of quality of frozen fish or that of frozen blocks or fillet or crumble seafood products remains much more difficult. Volatile amines are characteristic molecules responsible for the odor of fish stored for several days after the catch. They are commonly used as criteria for assessing the fish quality. Total volatile basic nitrogen, or TVB, represents the sum of ammonia, tarmethyl or trimethylamine, and legal requirements have been established for several fish species concerning TBV content. However, TBV content cannot be used as freshness indicator since it remains unchanged during first days after catch, after first days of ice storage. TBV nitrogen, therefore, is rather an index of spoilage and not an index of freshness. Moreover, volatilamines determination is also affected by several factors, such as volatilamines are leaky compounds, and storage of fish and slurry ice provoke the loss of volatilamines that are washed away with water, and it also depends on fish species. Nucleotides. Pure nucleotides issued from ATP degradation provide an excellent indicator for freshness evaluations for several reasons. Nucleotides are present in all living organisms and provide therefore universal indicators for freshness. The process of ATP catabolism is well characterized and documented by numerous scientific articles. And ATP and ATP catabolites are present in muscles in high concentration that considerably facilitates their quantification. These metabolites are thermostable, and the several HPLC-based methods have been established for accurate measurement of ATP degradation products in muscle. This method provides reliable results with only drawback. They are time-consuming and require expensive equipment. In living cells, ATP is constantly resynthesized by mitochondrial respiratory chain. Once animal is slaughtered, respiration stops, ATP cannot be resynthesized anymore, and it takes less than 1 day for ATP to degrade into AMP, several days to form inosine, and then the weeks to be converted to hypoxanthine. The concentration of pure nucleotides in fish meat or seafood meat provides several information on the quality of these products. Freshness, the higher is AMP content, the fresher is the product. Flavor property, the presence of IMP guarantees the taste and attractivity of the product both for human and animals. Nutritional quality, since these pure nucleotides present in food are reutilized by immune cells and cells of intestinal epithelium. And bacterial alteration, since the presence of spoilage bacteria result in increased hypoxanthine level. Our precise freshness assay kit is first method based on enzymatic conversion of IMP, inosine, and hypoxanthine into NADH that specifically absorbs at 340 nanometer. Once converted to NADH, each nucleotide is quantified by measuring absorbance at 340 nanometer using microplate reader. The kit includes 3 enzymatic mixtures, cofactors, and is provided with microplates pre-filled with nucleotide standards. All enzymes and cofactors are provided in stable lyophilized form and can be shipped without dry ice or cold blocks. The rate of enzymatic degradation of ATP depends on the temperature of storage. Freezing stops ATP degradation process. Therefore, the measurement of ATP catabolites in frozen fillet can reveal their freshness, which is often difficult to evaluate by other methods. On the other hand, a thermal treatment of cooking stops ATP degradation process while leaving ATP catabolites intact. Therefore, the content of nucleotides on cooked, canned, or dried products can be also used as a freshness indicator for raw material used before thermal treatment. The procedure of nucleotide measurement with precise freshness assay kit includes 4 major steps. Quantitative extraction of nucleotides is achieved by simple boiling of samples in provided extraction buffer. Filtered extracts are dispensed into the walls of microplate, followed by addition of enzymatic mixtures. During 30 minutes of incubation, added enzymes are specifically and irreversibly convert nucleotides into NADH, and at the end of incubation, the absorbance data are read with microplate reader at 340 nanometer. The possibility of using ATP catabolites for freshness evaluation has been demonstrated for the first time by Saito and colleagues more than 50 years ago in 1958, and further confirmed by numerous scientific publications. However, wide application of nucleotides for quality control in food industry was hampered by the absence of rapid methods for accurate nucleotide quantification and the variations observed in the rate of ATP catabolism between different species. So, freshness assay kit is first microplate assay that allows the measurement of ATP catabolites in 22 samples in parallel. This convenient microplate format offers an opportunity for systematic characterization of ATP degradation rate in a wide range of fish species. And using microplate format allows to study the effect of numerous storage conditions such as temperature, preservatives, packaging on freshness evaluation. Next slides will be focused on the results obtained with precise freshness assay kit. And first example shows the changes in inosine, shown in green, and hypoxanthine, shown in red, content in cold smoked salmon during storage. High blood pressure or hypertension is a major health problem that affects 1/3 of adults in the world. So reducing salt level in foods is an ongoing process. However, reduction of salt in foods remains a major challenge for food industry because salt was historically added to the food to enhance its shelf lifetime. So while reducing salt content, particular attention should be paid to maintain the freshness of these new products. So the effect of new preservatives on storage of cold smoked salmon was started by following inosine and hypoxanthine, inosine degradation and hypoxanthine accumulation using freshness assay kit. And as shown by this slide, this method provides an objective and scientific way for characterization of quality changes during storage. In particular, these results show that shelf life time was enhanced by 14 days for this particular preservative. Seafood processing generates a large amount of byproducts, especially sales, seafood processing and preparation of fish fillet. Big part of them is used to produce fish meal to satisfy growing demand of aquaculture. With continuing growth of the aquaculture industry, more attention is given to fish welfare since it has significant impacts on fish health and its resistance to diseases. Decrease in the resistance to diseases is a direct consequence of immunosuppression that can be induced by several factors such as stress or overpopulation during aquaculture. Numerous scientific articles have reported that high level of nucleotide in fish meal may enhance growth of fish or shrimps, strengthen immune system, increase survival rate, and finally helps to avoid the use of antibiotics. The level of pure nucleotides in fish meal was measured using precise freshness assay kit, and as shown by the graph, all three studied samples are highly rich in IMP, which is predominant product, meaning that this fish meal was prepared from freshly caught fish. High level of Inosina Monofosfato guarantees the attractivity of this fish meal for fish feeding, and total content of pure nucleotides observed in all samples is high, in particular in sample 3, and is sufficient, according to literature data, to provide immunostimulatory and probiotic effect on fish health. Next example concerns pet food. The pet food ingredients, for example, meat and chicken byproducts, are subject of rapid decay by microbial growth and autooxidation. So the freshness of pet food ingredients can affect considerably the taste of the resulting and attractivity of the resulting products. Off flavors caused by chemical or microbiological deterioration of the ingredient can result in food refusal, since animals rely on their sense and taste to differentiate safe foods from those which taste bad or may contain toxic substances. This slide shows the results of freshness analysis of pet food final products derived from two sources. High level of inosine observed in left panel indicates higher freshness of ingredient used for this preparation and suggests better attractivity of these products for the pet. Last example concerns seafood byproducts such as fish skin, which also are generated by the seafood processing industry. These byproducts contain a lot of valuable bioactive molecules such as marine collagen, used for pharmaceutical and cosmetic industry. Because of the presence of bacteria on the surface of the fish skin, the spoilage of fish byproducts is very fast and results in the degradation of biomolecules with high added values. The content of inosine and hypoxanthine in fish skin samples was measured using precise freshness assay kit and compared in 3 different samples This analysis has allowed to identify most spoiled sample, which is second, with lower inosine and higher hypoxantin value, in 1 hour. So this is all that I would like to say.
Thank you, Larissa. That was very informative.
Yeah. Thank you.
So BioTek collaborated with NovoSib to undertake automation of the assay for higher throughput analysis in 96-well plate format. The assay reagent and sample transfer were automated to offer increased throughput options in 96-well format, and with the goal of providing continuous liquid handling for one up to any number of plates while minimizing consumables and reagent use. Monochromator detection of NADH was validated at absorbance 340 against comparative filter-based microplate and cuvette detection methods, and sample prep optimization was undertaken to develop a method that quickly recovers larger sample recovery volumes common to higher throughputs while remaining compatible to diverse sample types. So automation has a number of benefits. In addition to instrumentation having specifications for sensitivity, specificity, linearity, and other performance variables, parameters can be pre-configured, offering a defined scope of performance. This combination lends ease to documenting standard operating and validation procedures, leading to reduced process variation. There are often built-in audit features or file or other runtime signatures for traceability and accountability. Instrumentation can be customized, offering opportunities for flexibility and versatility. There are usually built-in or documented maintenance procedures, warranties, and or service plans which give consistent care of the instrumentation, adding to its reliability. There are a number of price points available for instrumentation, offering economically realistic options. It also can provide known and repeatable time to results. It can reduce technician variability evident in manual methods, and it offers screening and analytical capabilities for a preventative feedback loop. The small footprint available on the instrumentation we will highlight today is also advantageous when there's limited bench space. These instruments also fit under standard 36-inch laminar flow hoods. So our featured instrumentation includes our precision microplate sample processor for automating the transfer of assay reagents and sample to the assay plate. That's on the upper left. On the upper right is the Epoch microplate spectrophotometer. We use that with the Gen 5 software for detecting the NADH at 340 and doing the data reduction for calculating percent freshness values of the nucleotides. For even higher throughputs than individual 96-well plates, the instrumentation is fully compatible with the BioStack microplate stacker. These stackers make robotic transfer seamless between the instruments. And we also have the opportunity to highlight our new Epoch 2 with touchscreen and Gen 5 software reader. This reader expands some of the features of the Epoch 1. It offers a front-loading cuvette holder, which is convenient for running this assay in cuvette format. It also has onboard linear and orbital shaking. And as seen by the picture, there's a built-in tablet that is preloaded with Gen 5 software and uses a touchscreen interface. So the instrument validation and sample prep optimization happened in 4 stages. There was monochromator detection and automated liquid handling validation using NADH standards. We then moved to optimizing the liquid handling using assay reagents that reflected running the assay in real time. We optimized sample prep methods for higher sample recoveries common to higher throughputs. And then we did some challenge testing to validate the applied automation and monochromator detection. So the first set of data here shows the linear range of the Epoch monochromator for sensitivity and specificity to NADH. 15 standard points of NADH were prepared in potassium hydroxide. They were provided by Novosiv to help calibrate the reader. We tested 2 runs on 6 readers for a total of 4 plates of interassay data. We then plotted the data we received from the Epoch against comparative filter-based microplating cuvette data and the expected data as provided by the supplier of the NADH, and that's shown on the left. The linear range of the Epoch at absorbance 340 using the NADH standards equated to 0.515 to 2.50 OD, or somewhat greater than 50 up to 620 micromolar. However, even at lower ODs, the distinct peak of NADH is nicely illustrated using the spectral scan capabilities of Epoch as programmed within the Gen 5 software. NADH standards and 0 micromolar blanks of in potassium hydroxide were read from 300 to 400 OD in 10 nanometer increments. These points were run in duplicate, and both points for each standard in the blank are plotted on the spectral scan data on the top and bottom right. Duplicate points track with high correlation, and even at 0.05 OD, the 12 micromolar standard shows a distinct peak at 340 compared to the blank, which is shown at the top right. This pattern repeats continuously as shown for the 27 to 150 micromolar standards in the graph at the bottom right. Final maximum OD at 340 for the cuvette method used to calibrate new sample matrix volume dilutions is 1.3 to 1.5 OD, corresponding to 250 to 500 micromolars of NADH, and this is well within the linear range of the Epoch monochromator results for NADH. Automated versus manual pipetting was validated also using a 15-point standard curve of NADH. This data is the result of 2 separate runs of 2 plates each over 2 days. Of note, the automation has higher precision and is calculated by a CV of 0.821% compared to 1.2% of the manual method. And although accuracy is slightly lower for the automation, it is more consistent than the manual method. To test solubility and stability, influence on background signal, effects of reagent viscosities, and to determine optimal reagent usage based on actual final volumes, instrumentation was validated using the assay reagents. This was done over 3 runs using 6 plates by 2 different operators at dilutions of 1, 2, and 10x of each enzyme mix. Results for 2 of the enzyme mixes is shown at the top. Reagents are stable when solubilized, even under elevated temperatures within the hood where automated liquid handling occurred. Results are correlative between 2 operators, one of which was completely unfamiliar with the assay method. Differences in the automated and manual method precision was correlated to outliers resulting from insufficient dead volumes highlighted by the experimental design. This was observed during the workflow. The experimental design was therefore changed to reflect running the assay in real time. To maximize reagent usage, the loading layout and dead volumes were adjusted. This optimization provided a benefit of reducing consumable use while providing an increased dead volume as further prevention against insufficient reagent volumes during pipetting. A mock throughput run of 6 plates was done to verify the optimization step using a final continuous liquid handling throughput model, resulting in 0 outliers. Because the assay was developed for lower throughputs in cuvette format, an alternative sample clarification step conducive to automation at higher throughputs was required. This would be one method that provided fast one-step, one-pass filtration while recovering higher sample volumes was needed. Homogenization, centrifugation, and a variety of filtration methods were validated. At the top left are results from testing homogenation and centrifugation with and without added single-layer 0.45 micron vacuum filtration. A clarification method not requiring homogenization or centrifugation was desired. Although optimal results were obtained from centrifuged samples that were either homogenized or not, all methods presented workflow challenges. So in the case of homogenizing the samples and centrifuging the samples, there were a number of transfer steps that were required that made the workflow less efficient. And in the filtration step, the 0.45 micron required 3 separate passes before final clarification was obtained, and this was validated by centrifuging the exudate until no pellet formed. Since the non-homogenized filtration method provided results correlative to the other results, a number of different filtration methods were validated. A final method of choice was a multi-layer glass 1.2 micron and 0.2 micron disc filter attached to a syringe. This method was verified using an assay run of a volume dilution on raw, frozen, peeled shrimp. Results on the lower right show that at higher concentration of sample, nucleotide recovery and detection were optimal. Since the volume dilution of 2x had been pre-calibrated as optimal for this matrix by NovoSib, the one-step multi-layer syringe filtration method was adopted for all further testing. So we're going to show a video now of the final workflow that we used, and we're going to play that.
Yeah.
So add 2 to 5 grams of sliced muscle tissue to a 50 ml conical tube followed by assay extraction buffer. Boil the samples for 20 minutes. Meanwhile, prepare assay reagents. Add cofactors 1 and 2 to assay reaction buffer and mix well by gentle inversion to ensure complete recovery. Enzyme mixes must be freshly prepared for each assay. Dilute enzyme mix 1 with 8.4 ml of the reaction buffer containing the 2 cofactors. Then transfer 5.6 ml of enzyme mix 1 to enzyme mix 2. Reconstitute cofactor 3 into enzyme mix 2. Since the rate of enzymatic reaction depends on the temperature, it's important that reagents are at room temperature before these solubilization steps. Finally, transfer 2.8 ml of enzyme mix 2 containing all 3 cofactors to enzyme mix 2. Load 330 microliters per well of each reagent to a holding vessel in a 2x4 pattern. Reaction buffer is loaded to rows A and B for blank measurement, enzyme mix 1 to rows C and D for IMP measurement, enzyme mix 2 to rows E and F for IMP and hypoxanthane measurement, and enzyme mix 3 to rows G and H for IMP, hypoxanthane, and inosine measurement. Transfer samples to an ice bath for at least 5 minutes to cool, then prepare precision by loading 2 sets of tips, up to 2 assay plates, and the reagent holding vessel to the deck. Using a 10 ml syringe, aspirate sample exudate directly into the syringe and fix to a volume from 2 to 5 ml. 2 ml is the minimum required to accommodate overage volumes for automation, but up to 5 ml can be loaded to each column, providing a larger sample pool if desired. Attach a multi-layer disc filter to the syringe tip and dispense the sample directly into one lane of the holding vessel. Repeat for each sample, then load the holding vessel to the deck of the Precision and begin the program. Using the 8-channel pipetter, Precision mixes, aspirates, and then dispenses 100 microliters per tip from the sample vessel to the assay plate. Column 12 of the assay plate is reserved for pre-filled assay controls that are reconstituted from extraction buffer loaded to the holding vessel. 100 microliters per tip are then mixed, aspirated, and dispensed from the first column of the reagent vessel to the assay plate. 3 cycles are aspirated and dispensed from each column of the reagent holding vessel before moving to the next column. 4 columns fills a 96-well plate. At the end of the run, Precision will pause, allowing removal of the assay plate and supply replenishment for subsequent plates. Read the plate at 340 after a 20 to 60-minute incubation at room temperature. Raw ODs and all data analysis are easily viewed using the Gen 5 drop-down menu. Calculation of relative nucleotide content using the formula provided in the assay kit insert are built into a sample file available from BioTek and can be easily edited to suit individual applications if necessary. Tips and holding vessels are autoclave compatible for reuse, and the ease of a front-loading cuvette holder on our new Epoch 2 expands assay workflow options further. So that's the workflow that we used to undertake some challenge testing of the automation. We used a variety of sample matrices and experimental conditions, and it should be noted that although the enzyme kinetic reaction of the assay stabilizes after 20 minutes at room temperature, we used a 1-hour incubation during challenge testing based on previous versions of the kit insert. The samples we used in our challenge testing included raw, frozen, peeled, whole shrimp, and that was 2 brands. One was imported farmed and one was American wild. We also used canned, wild-caught, tiny North Pacific, served in water and citric acid. We used a live North Atlantic female chicken lobster and also to compare against a raw, frozen North Atlantic tail meat product. For the salmon, we used 2 brands of packaged smoked salmon. We had a 6-piece sushi packaged on the purchase date from a retail grocery sushi bar, and 6 pieces of sashimi that came from a restaurateur made to order. For the experimental conditions, we ran a 90-day time course on the frozen shrimp. We also compared brands and different matrices of the same species using the freshly slaughtered lobster and frozen lobster tail, the 2 brands of packaged smoked salmon, and the 6-piece sashimi versus the 6-piece sashimi. The 6-piece sushi compared to the 6-piece sashimi. We also looked at the effects of storage methods on freshness. So we used a variety of samples at 5 different storage conditions, including -80, -20, 0 to 4 on ice, 0 to 4 not on ice, and at room temperature. When not tested on the day of preparation or purchase, samples were pooled and divided into 3-gram sample lots and vacuum sealed. Here we give a representative sampling of results published in the application note. So for the 90-day time course on frozen shrimp, here you see the blue is the IMP, and it's decreasing over the time. The red is the hypoxanthine increasing over time. And the inosine has a parabolic shift as the IMP degrades to inosine and then the inosine degrades to hypoxanthine. The experiments were carried out on 2 lots of assay reagents. Day 1 and 45 were done on lot 1 reagents, and remaining time points were done on lot 2 reagents. Product was labeled with a use-by date 6 months from the date of purchase. The range of time between test points varies from a high of 45 days between tests to only 4 days. This is reflected in the graph as the highest change of nucleotide percent correlates to the longest time between test points at 45 days, and the least change is where test points were only 4 to 10 days apart. So here's some representative data of the effects of storage conditions on relative nucleotide content in the frozen shrimp left versus canned shrimp on the right. And this is measured by percent change over 10 days from 3 time points. Day 1 was the purchase date of the products. Canned shrimp is pre-cooked and preserved in water and citric acid. It shows the most stable freshness, although as expected begins to deteriorate when kept at any storage conditions except -20 over 10 days. Although the frozen shrimp begins to deteriorate at all storage conditions, even at 20 degrees C. It should be noted that day 1 testing of this product corresponded to 78 days post-purchase date. As expected, the highest increase of hypoxanthine is detected when previously frozen shrimp is kept at 0 to 4 or room temperature over a 10-day period. This is shown by the large red bars. The data suggests that time and temperature can be used to stress the ATP degradation pathway in muscle tissue, manipulating freshness conditions to optimize product taste, texture, shelf life, or other parameters. For the fresh and frozen lobster, the data here is compared and also illustrates how time and temperature can influence freshness parameter concentration. So in the top graph in the upper left, you see the fresh tail, fresh joint, and fresh claw. And they were tested right after slaughter to see if the lobster can be tested using the claw or joint muscle, saving the tail muscle for final product. But as you can see, the results of the experiment actually showed very low levels of IMP recovered in freshly slaughtered lobster compared to the frozen lobster tail. So it was hypothesized that 1 hour post-slaughter at room temperature was not enough time to reach relevant or optimal IMP levels. So we ran an experiment to prove this hypothesis in the lower right. And as you can see that after the freshly slaughtered lobster was kept for 24 hours at 0 to 4 degrees C, IMP increased dramatically. You can see circled in the middle of the graph. When it was kept on ice for 72 hours, it also shows a dramatic increase in IMP. So we screened 5 samples of sushi and 5 samples of sashimi. Both were salmon, but from different suppliers, and they show fairly correlative results. Although the IMP is low, the hypoxanthine hasn't increased dramatically. For the sashimi, an outlier was detected for 1 piece. This result highlights the characteristics of a good quality test, which is the ability to screen multiple number of samples simultaneously while receiving analytical data that can confirm conformance to specification, while providing information used to infer corrective action. In the case of these products, the U.S. regulates that fish served as sushi or sashimi be previously frozen, but effects of thaw conditions on freshness levels could render increased variability among the pieces. Data showing an inconsistency between freshness of previously frozen fish could be used, for example, to insist in providing required % contact for samples using standardized thaw condition procedures. At the end of the challenge testing, and I just showed a representative sampling of that data, we came up with a Gaussian distribution of expected automated assay performance. On the right hand of this graph are the controls that are pre-filled in the microplate, and the blue corresponds to the IMP positive control, the green to the inosine positive control, and the red to the hypoxanthine positive control. On the left are the results of the negative controls and the blank. The blank, this is the black line is the raw data for the blank values, and the blue and green lines and red dots are the data for the negative controls for hypoxanthine, inosine, and IMP. So we calculated a limit of blank, a limit of detection, and a limit of quantification using a 19% error. So the limited bank is 0.308, the limit of detection is 0.515, and the limit of quantitation is 0.612. The distribution graph shown here was done on replicates of 32 for the blank, 15 for the negative standards, and 13 for the positive standards. They represent 13 plates of data on 2 lots of kit. So in conclusion, The NovoSib Preci Freshness Assay workflow is amenable to automation in 96-well microplate format, and enhanced throughput options are offered by BioTek instrumentation suitable for a broad range of customer needs. The challenge test data suggests that automated procedures offer an effective tool for food quality control and research environments. Thank you. For more information, you can contact BioTek at www.biotek.com. And for assay information, you can contact NovoSib at www.novosib.com. Remaining information is on the slides. Thank you, and I'm going to turn it over to Lindsay now.
Thank you very much for joining us today, and thank you to our speakers for their presentations and for taking time out of their day to join us for this live event. We will email this recorded event to all participants. If you have further questions for our speakers or would like more information on BioTek's products, please visit our website. In addition, please check out our blog, like us on Facebook, and follow us on Twitter to hear more about BioTek webinars or to learn more about our products and how they can help you. Again, thanks for joining us.